Hello and welcome to my talk. I'm Katharina Schmid from the Eqtl Lab, which is part of the Computational Health Department from the Helmholtz Center Munich. Today I will talk about design and power analysis for multi-sample single-cell transcriptomic experiments. For this we developed an analytic power analysis method called scPower. As you all know for sure, single-cell RNA-seq has led to many new exciting analysis possibilities. Here we want to focus on its application for multi-sample experiments where we compare differences in a population. Our group is especially interested in Eqtl studies. That is short for expression quantitative trait locus and describes genetic variants associated with gene expression. If you use now single-cell RNA-seq instead of bulk to quantify the gene expression, you can capture cell-type-specific Eqtls which might be masked in bulk. A study that showed this very nicely in PBMCs was performed by the Frankel Lab. That's a picture from it, where they depicted here a monocyte-specific Eqtl. So you see no association in all cells. That would mean that you see no association in bulk, but you see a clear association in the monocytes, especially in the classical monocytes. The same kind of analysis can also be done in general for any kind of differential expression analysis, where we compare differences between groups in a population. That's here a plot from a study from the Lung Cell Atlas where they compared asthma patients and controls and also found several genes with cell-type specific differential expression. So as you see, single-cell RNA-seq provides very valuable information for multi-sample experiments. However, it requires to measure single-cell data for a whole population, which can be of course quite cost-intensive. So it's here especially important to perform a power analysis before and identify the optimal experimental parameters for the experiment. And that's exactly the goal of our tool. That's a general overview of our approach. It's an analytic method, which means it's very time and memory efficient, and we developed it in a user-friendly and flexible way with many adjustable parameters. It estimates the power here in blue in the middle, dependent on the three main experimental parameters: the reads per cell, the cells per sample, and the number of samples. Additionally, also a number of priors play a role here in green, which you can get either from previous experiments or based on assumptions, for example the effect size. And then we have additional user-definable parameters. So first we have the cell type detection probability. That's the probability to detect a certain number of cells of our cell type of interest. For this already several interesting methods exist, so it's a prerequisite, but we will focus on the second part today, the overall detection power. That's the probability of finding DE and Eqtl genes in our cell type of interest. So this overall detection power can be split into two parts: the expression probability and the DE and Eqtl power. For the expression probability, that's the probability to detect a gene in the dataset. And as single-cell RNA-seq data is much sparser than bulk, that's here especially important, as we will lose part of the genes already because it will not be detectable in the dataset. And then if we know that a gene is detected, we can look at the DE or Eqtl power itself, which is the probability to define significant differences between the groups. Both parts depend on the mean and dispersion of the gene, which we will model with our tool. And to explain this, I will start showing you how we build our expression probability. So to start with this, we first looked at the number of expressed genes in different single-cell datasets. We define a gene as expressed if it has a certain number of counts in a certain fraction of individuals, and both thresholds can be set flexible dependent on the user. Here we say it needs to be expressed in more than 50% individuals. In the first column, looking at a count threshold larger than 10, and in the second at a count threshold larger than 0. You see in the first row the results for our own PBMC dataset that was generated by the Max Planck Institute of Psychiatry in Munich. You see that the more cells we have from our cell type, the more expressed genes we find. Additionally, also the read depth is important. That's here shown by the different colors. So the higher the read depth, the more expressed genes we detect. Each point represents a different cell type, and we will also model this in a cell type specific way, as the different RNA content will lead to different number of expressed genes dependent on the cell type. In the second row, we show the results for another data set that's publicly available from Chang et al. That we will later use to validate our model. As you've seen, we are interested in the expression per cell type in person and not on the cell level. So we build our model on the so-called Pseudobug approach. This is, combined with classical DE and Eqtl methods, currently the best performing method for multi-sample single-cell transcriptomics experiments. We start with our count matrix of cells times genes, where each cell is annotated to a cell type and donor. And then by aggregating the counts per cell type and donor, we generate this three-dimensional pseudobug matrix. If we want to estimate the expression probability of a certain gene in one cell type, we first go back to the single-cell count matrix. We model the count distribution of a specific gene in a specific cell type using a negative binomial distribution, which is well established here. So we have a mean and a dispersion parameter to describe it. And the pseudobug, because it's a sum of negative binomial distributions, we have again a negative binomial distribution, and both parameters are shifted by the number of cells. With this, we already get the first of our main experimental parameters into our model. Using this information, we can build already a first expression probability for this gene, as a combination of a cumulative binomial and a cumulative negative binomial distribution. Here, the number of samples, the second experimental parameter, is important. But you see that the redepth is still missing, and in the following we will also incorporate this. Here, in this parameter space, we have the mean and dispersion parameter of the gene as additional information. However, we are in the end, of course, interested in the expression probability for all genes, so this means we have not only one negative binomial distribution, but one for each gene, which generates a mean and a dispersion vector. We abstract the dispersion vector using a mean dispersion curve. That's a very classical approach already used, for example, in the ECCB. And then if we look at the mean vector, we see that all genes within one cell types follow a gamma mixture distribution with two gamma components and one zero component. So we model all means according to this distribution. And then to incorporate now the redepth as promised, we subsampled our count matrix and saw that for different redepths, our gamma mixture distribution is shifted. So we parameterize this gamma mixture by the redepth. And with this, we have finally now our three main experimental parameters in the formula, and we need a few additional hyperparameters to describe these gamma mixture distributions dependent on the cell type and the mean dispersion curve. And this model works quite nicely. That's the curve that you've seen before. So the solid lines are the observed counts, and now we predicted the number of expressed genes with our model, and the result are the dashed lines. And you can see that it matches very nicely not only for our own PBMC dataset that we used to generate these gamma mixture distributions, but also for the unseen public available dataset. So also here, although it was measured in a different lab with different redepths and a different number of samples, we can predict the number of expressed genes very nicely, which shows how robust our method is. So now that we are confident in the first part, we can look at the DE or Eqtl power. For this, we can use methods that are already established for bulk, given that we know the mean and dispersion parameters, which we can get from our expression probability model. And then we need to incorporate additional prior information of the effect sizes and expression rank. This we take from FAC-sorted bulk RNA-seq studies to get cell type specific estimates. And for the effect sizes, so these are the beta values. In case of Eqtl studies, it would be log-fold changes for an DE study. And the rank describes how are our Eqtl or DE genes distributed among all expressed genes from the expression level. For example, if they would only be among the highest expressed genes, we would not need to capture so many genes. However, we see that most of the times the DE or Eqtls are very uniformly distributed among all expressed genes, so it's important to detect as many genes as possible. We additionally have a significance threshold that we correct for multiple testing, given the number of expressed genes that we get from our expression probability model, we have here different options using either Bonferroni or FDR. If we combine both to the overall detection power, we can build a product of both because they are both conditionally dependent given the mean and dispersion parameter. Here you see example calculations for one DE and one Eqtl study, again split into the 3 parts. The expression probability depends mostly on the number of cells in the x-axis. So the more cells, the more genes we detect, as shown already in the beginning, while the power itself then also is strongly influenced by the sample size, especially here in this Eqtl case. And then we have the overall detection power as a combination of both. We validated our method by comparing it against different simulation-based methods, here PauSimR and Muscat, and each point in the plot is now a different parameter combination. We looked again at the expression probability, the DE power, and the overall detection power, and for all 3, our results matched very nicely the simulation-based studies. The big difference is the runtime and memory. Because our method is analytic, it takes less than a minute to calculate all these different parameter combinations, while it takes multiple days for both simulation methods. And also the memory can be an issue if you want to simulate a large population, as it's often required for Eqtl studies. So the strength of our method is definitely to compare a lot of different parameter combinations. We extended this further by also incorporating the budget. A realistic use case is often that we have a specific experimental budget that we can spend, and we want to look what parameter combination is the best, given this budget. For this, we incorporate also a cost function, again dependent on our 3 main parameters. And this gives us now a grid of possible parameter combinations. We have here the number of cells and the read depth, and the third parameter, the sample size, is then uniquely defined given the other 2 and the budget. To look here in this case, we get the best out of the money if you take a low read depth and a relatively high number of cells. To look a bit more in detail at the trends, if we have a fixed read depth, here that's this column, we see in plot C, the more cells we measure, the higher is our expression probability, but measuring more cells means also that we can only measure less samples, as we have this budget restriction, which will lead to a decrease of the power. And so at some point also the overall detection power. And a very similar trend is also visible for the read depth. We did this evaluation now for different budgets and also for different priors, so high effect sizes versus low effect sizes and so on, and also for different single-cell platforms for 10x, DropSeq, and SmartSeq2. So our tool is in general applicable for any kind of droplet or plate-based single-cell method. And what we see for a lot of the evaluations that we did, that in many cases shallow sequencing of a large number of cells is beneficial. But the exact optimal parameter combinations depend a lot on the specific settings you have. So if you got interested in testing it for your next experiment, I would really recommend you to test our tool yourself. For this, you can use these nice website that we set up, or you use directly the R package that we have here on GitHub that gives you even more flexibility. And with this, I come to an end. I want to thank my supervisors, Matthias and Fabian, and Barbara, who set up the nice website. And I also want to thank the people from the Max Planck Institute of Psychiatry and the Institute of Diabetes and Regeneration Research who generated the dataset for us. And thank you all for your attention.